Good morning, everybody. Good morning. My name is Robin Frankel. I'm chairman of the Friends organization, and I want to welcome everybody here today. And we promised that we would get everybody out by 9 o'clock, so I'm trying to stay on schedule. Our speakers today are going to be in this order: Dr. Danforth, who's chairman of the board; Sam Fiorello, who is the center's chief operating officer, senior vice president, and president of the Bioresearch and Development Growth Park that we call Bridge Park next door; and Dr. Phil Needleman, who is a trustee and serving as our interim president of the center. Following their remarks, there will be a time for question and answers, and to make sure that you're heard, be sure that you've got a microphone in order to ask a question, and there will be people circulating with mics. I also want to tell you a little bit about some upcoming activities for the Friends Committee for the Center. We've got a conversations program coming up on February 25th, and the topic is going to be Women in Science. And then on Thursday, March 11th, Ira Flato from Science Friday is going to be our special guest for a program. Both of those begin at 5 o'clock in the evening, and we hope you'll make plans to attend both of those. They're going to be really special events. If you're not a member of the Friends organization, I have to make a plug. And when you're a member of the Friends organization, not only do you get invited to all of these special events that occur throughout the year, but you're also helping to support the mission of the Danforth Plant Science Center to feed the hungry, to preserve the environment, and maintain the relationship of St. Louis as a world center for plant and life sciences. So, without further ado, I want to introduce Dr. Danforth and start the program. Thank you very much. I'm raising the bar.
Thank you. Thank you. I was about to know. Thank you. Thanks, Robin, very much, and thanks for setting this at just the right height for you or for me. And thanks to Robin for doing such a good job in leading our Friends group. I'd like to welcome all the friends and supporters of plant science. It's good to have you here. Appreciate your coming to the 2010 report on the state of our center. Last year, I called you shareholders, a title you'd earned by giving us your encouragement, your enthusiasm, your hard work, and your treasure. We're grateful to you, and I am grateful. We owe you an annual accounting, and this is it. We want to tell you the bad as well as the good, and you've earned the right to question us, and I hope you will when we finish. In 11 years, we've come from an insubstantial dream to a position of national and international leadership. I'm personally thrilled and have been thinking about how and why. We have had generous friends, hardworking volunteers, outstanding scientists, dedicated administrators and staff. We've had a great leader in Roger Beachy, and do now in Phil Needleman. But there's something else as well, something that will be our theme today. We are at the right place at the right time. The environment, except of course for the economy, has changed in our favor and is now supporting rather than impeding us as it was in the early days. We've been trying to plan well, work hard, and climb upward. The change is now that the environment is like an escalator speeding our ascent. What we have been building here has prepared us to work with others as they catch our vision and see the same needs and opportunities that we see and have been seeing. I'd like to think that our foresight has contributed, and maybe it did, but luck always plays a part in human affairs, and we've been lucky. Maybe, as Pasteur said, chance favors the prepared mind. We have been prepared. Today you'll be hearing from Phil Needleman and from Sam Fiorello. They'll report on the doings and accomplishments and how our efforts have been supported by happenings outside our walls. But first, a little history. We started the center because we believed that advances in the biologic sciences could be applied effectively to plants with benefits to humankind, internationally as well as locally. Second, we could do first-class basic research and achieve practical results. Third, no not-for-profit institution was making the most of the opportunity. And fourth, St. Louis was the place to try to do it right. We realized there were challenges. Agricultural science had been a low priority for over 30 years, low national priority for over 30 years. Important federal programs had been poorly managed and underfunded, especially compared to medical programs, and many thought we were on the wrong track. But the world changes. Who would have thought that today we would have had a National Institute for Food and Agriculture passed by Congress and enthusiastically supported by the present administration, or that Roger Beachy would be its leader, or that the administration would recommend for next year a 62% increase in the key budget in this economic climate. Who would have thought today that the Gates Foundation and the Howard Hughes Institute, two of the nation's largest private funders, would be embracing agricultural research? Who would have thought that the Department of Energy would have hit our sweet spot, or that two of two Algae Center grants would have come to St. Louis, one to Washington University and one to our center? Who would have thought that the national competition, we would head a consortium of institutions, and here are the institutions, many well-known large institutions. We are the lead agency of these institutions on a $44 million grant for algal research, and that Dick Sayre, a scientist here, would be the lead scientist, or that all of these institutions would be included. We could not have known that we would convince a private developer to build a building for commercial enterprise on our north 8 acres. We might have lost heart after some false starts we had, but now we have a wonderful development partner, and Sam Fiorello is making Bridge Park a vital part of our community. But maybe we should have expected good luck, because we've had good luck from the start. Monsanto saw the world as we did and has supported us generally, as has the state of Missouri and the Danforth Foundation. We recruited a visionary leader from the West Coast, who in turn recruited a marvelous scientific team. You, our friends, joined us, and annual support, starting from 0, has grown to an amazing figure that I will not disclose because Sam is going to present it shortly. But when he does, you will see that we would not be where we are without you, our hardworking and generous friends group. Jack Taylor, John McDonnell, Ollie Langenberg, Mel Bali, David Kemper, and all our friends saw the vision and responded with great generosity. Roger traveled the globe, and people began seeing and believing. Our field has gone from being neglected and thought unimportant to important for improving human nutrition and preserving the environment. Another piece of good fortune with Roger's appointment, we could have been without a leader, but fortunately we had on our board one of the nation's great biologic scientists and scientific leaders, Phil Needleman. He's brought us new ideas on how best to conduct science and advance us much farther than we would otherwise have been. Now, a brief report on how we're going about the search for the president. We have a committee, and here are the members, with Roy Vagelos as the chair. of the group. We asked for proposals from search firms to help us with this undertaking. Roy screened 20 or so by phone and then settled on 4 to recommend to the committee. The best presentation that any of us had ever heard from a search firm was from Eileen Nagel of Russell Reynolds. So we employed her. We sought advice from 2 groups on and asked about the most promising opportunities in plant science in the future and the strongest candidates to be our leaders. We first asked the scientists in this center and then made a list of 23 of the nation's leading plant scientists and asked them to be consultants and asked them the same questions. None of the people we asked to help turned us down, and there was pretty close agreement, especially close agreement between inside and outside scientists. We then selected a few of our advisors and asked each of those individuals to meet with us for further discussion and advice. Now, in that way, we could get to know people as consultants, and they could get to know us without sort of putting them on the spot of being declared candidates. It's sort of a little nice way of going about our business. So searches never go as fast as one would like, so I'll have to report later on how we came out. People say, "How are you doing?" And I say, "I never know until the person has signed the document." So in summary, all summary, good fortune has led us to the right people and the right building and the right equipment at the right time. With your help, we've had the strength to reach a position of national leadership. Now it's our chance to play an even larger role than we had anticipated. As you listen to my colleagues, you will not be hearing that difficulties and tough challenges are behind us. On the contrary, you will likely gather, as have I, that new opportunities and important services bring more and more challenges than ever before. If we play in the major leagues, we have to work harder. Now, as you listen to Phil and Sam, think about running hard, but running up an escalator. Think about the great opportunities to serve the world and our home community. Now I will turn this podium over to Sam Fiorello. Great.
Thanks, Dr. Danforth. I'd first of all like to echo Dr. Danforth's words of welcome and thanks to all of you for being here this morning. Right place, right time, and prepared. As Dr. Danforth said in his remarks, this is not an original idea. Great minds and thinkers have been thinking this for many, many years. Today, unprecedented attention and resources are being allocated to ag science to develop potential solutions to some of our world's most pressing problems. Finding economically viable alternative fuels, discovering better ways of feeding the hungry world, and preserving our threatened environment. And we are at the center of these issues, and we are succeeding because we're prepared. We have developed an enterprise that takes advantage of the alignment of intellect and resourcing. At the Danforth Center, we are conducting great science, and we're using resources very efficiently. We are proud to announce that again last year, we outperformed our peer group, maximizing support for science while keeping our overhead costs down. In 2009, 83 cents of every dollar spent at the center was used to directly support our science. This versus a 77-cent industry average. We pride ourselves on our continual drive to add value and efficiencies into our organization, both serving as good stewards of our funds and providing the highest level of service to our scientists so that we will better achieve mission impact to improve the human condition through plant science. Here's but one example of the kinds of things we do in our pursuit of excellence. In 2005, we decided it was time to find a more streamlined system for ordering scientific supplies and equipment. With our accounting team and scientists working closely together, we invested in a newly created software system targeted for our type of organization. This may not be very sexy, but it's a real winner for both science and cost control. Scientists now have a web-based service with immediate online access to laboratory supply catalogs from hundreds of vendors, allowing the labs the ability to identify competing suppliers, which assures we're paying the best prices. PIs and their lab staff can now place orders electronically for supplies when they need them without any added work from the accounting department, saving time, effort, and hassle. Our initial investment in purchasing and implementing the new software was $130,000, but our new system saved major administrative costs on consulting and administrative charges that we simply got rid of. Almost $93,000 per year since we implemented the change. And the scientists love that they can get what they need when they need it with much less paperwork. One of the things that Roger Beachy deliberately set out to do when he built a scientific team here at the Danforth Center was to select scientists that had a demonstrated record of collaboration. His philosophy was that when scientists work together, they could not only produce better science, but also that that better science would make those scientists and the center better able to compete for outside funding. From the beginning, collaboration has been embedded in the DNA of the Danforth Center. Here are a couple of current examples. The first is the BioCassava Plus grant funded by the Bill and Melinda Gates Foundation and features 6 center scientists. The second, also featuring 6 center scientists, is a recently awarded biofuels grant by the Department of Energy. This level of intra-institutional collaboration is not the norm and has indeed enhanced the center's ability to win competitively awarded research funding. We are excited that grant and contract revenues continue to grow at the center. Grant and contract revenues have jumped from $2.1 million in 2000 to $12.5 million this year. This success is in a very competitive funding environment. For example, in 2009, the National Science Foundation award rate was 1 out of every 10 grant submissions. This is a real tribute to the terrific work of our scientists and their skills. Today, the average level of grant and contract award for researchers in the plant science field is roughly $350,000 per lab per year. These are no slouches, mind you. These are researchers at some of our country's finest agricultural institutions. Our scientists are doing far better than that. In fact, about 2.5 times that plant science average, or about $900,000 per lab. Certainly our scientists are at the right place at the right time. But it's their excellence in their scientific discipline, culture of collaboration, terrific infrastructure and back office support that makes them prepared to take advantage of that confluence of time and place. We continue to experience growth in unrestricted annual support. Dr. Danforth alluded to this, increasing from $400,000 not that many years ago, 2001, to last year, $1.5 million. Truly impressive. This impressive growth is a true tribute to you, our generous donors. It's also a tribute to Dr. Danforth, to Laura, and her entire development team. Of course, none of this would be possible if not for the great work of our volunteers, especially our terrific Friends Committee. Our Friends Committee helps with everything from planning events such as our conversation series and Plantasia to assisting in our phone-a-thons. They also serve as ambassadors, representing the goodwill of the center to all their friends and colleagues. Their efforts were enhanced in 2009 as a new 60-member Danforth Leadership Council was formed to help us further understand and connect with our St. Louis community. Even more than before, in 2009, our volunteers and donors rose to the challenge. In this case, the challenge was real, a donor stimulus fund provided by leading donors to encourage new donors to join the center and existing donors to increase their generous support of unrestricted funding. This unrestricted funding is absolutely critical for fulfilling our mission. These funds help give us the necessary leverage to attract the large grants and awards previously mentioned. With our recent success and growing needs for things like greenhouse space and scientific equipment, increased annual support will be key to ensuring that we continue to seize the opportunities before us. The new Bioresearch and Development Growth Park, which we refer to as a Bridge Park, is also succeeding. In It is succeeding because the Danforth Center began preparing for its creation from the center's very beginning. While research for knowledge and discovery's sake is important, it contributes to society even more when it is brought into the marketplace. And that's where Bridge Park will make a difference, helping entrepreneurs grow their research from good ideas into commercial successes. As a next-door neighbor of the Danforth Plant Science Center, Bridge Park complements our global vision. Last June, Bridge Park opened the first of 3 buildings that will ultimately encompass a 500,000 square foot campus and house over 1,100 high-paying jobs. It's a first-class facility developed by our terrific partner, Wexford Science and Technology. Already 2/3 of Building 1 has been leased, this in a tough economy. Tenants range from a leading ag tech company to an algal biofuel startup company, a prominent incubator for plant and life science companies, and an on-site workforce development and training program through the St. Louis Community College. More importantly, commercial success at Bridge Park will help fulfill the promise of ag science to bring a brighter economic future to St. Louis and Missouri. All of us are lucky to be part of the dawn of a new era for our region. St. Louis is not just becoming an important place for ag science worldwide, it's becoming the place for ag science. In addition to providing the vital components necessary to help companies further their commercial success, Bridge Park has already taken a leadership role in several important areas. With the NIDA Center for Scientific Enterprise, we created the Ag Innovation Showcase, a signature event which in its first year attracted nearly 200 attendees and established itself as a leading global annual event for agriculture's industry leaders, entrepreneurs, venture capitalists, and investors. The Ag Showcase will return to the Danforth Center this May. It promotes emerging technologies in ag bio, food and nutrition, biofuels, sustainable materials, and clean tech. Just last month, Bridge Park became the home to the St. Louis Community College Center for Plant and Life Sciences, a flagship workforce development and training program that operates right alongside emerging entrepreneurial ventures and world-class science of the Danforth Center. In addition to being a showcase for innovative job training programs in our state, the students are a valuable resource for companies in all stages of development. What a great opportunity for the students, new companies, the center, and our region. Also last month, Governor Jay Nixon chose Bridge Park as a setting to announce his plans to grow and attract more science and technology companies and jobs in Missouri. The Missouri Science and Innovation Reinvestment Act, or MOSIRA, has generated bipartisan support in the General Assembly and has good prospects in a tough legislative year. And Bridge Park has helped set a very high environmental standard for laboratory research facilities by earning a coveted Gold LEED certification for sustainable design and construction from the U.S. Green Building Council. We continue to look forward for growth opportunities at Bridge and are working on designs for the second building on the campus. Although we're at the right place at the right time and the accomplishments that we have made are significant, we still have much to do. We plan to continue to work hard and strategically to maximize the strength of the Danforth Center and Bridge Park to help make the world a better place by adhering to our threefold promise: to feed the hungry and improve human health, to preserve and renew our environment, and to enhance our region as a world center for plant science. We are well on our way because we've been prepared, and we continue to work to take advantage of all opportunities and luck that comes our way. I want to give a special word of thanks to Roger Beachy. As Dr. Danforth mentioned, Roger left his position as president of the center last October to serve as the first director of the newly created National Institute of Food and Agriculture. Although Roger has been at his new leadership role since October and is greatly missed, I think it a real testament to his leadership that the center still thrives in his absence. Founder and CEO of the Acumen Fund, Jackie Novogratz, said of great leadership, "The task of a truly great leader is to leave an institution that is better because of them, but not dependent on them." Although I miss Roger, and we all miss Roger, like Dr. Danforth said, we are so fortunate to have someone with Phil Needleman's skills, dedication, and intellect step in as our interim president. Phil is a terrific person to work with, and I've enjoyed learning new things from him on a daily basis. And finally, I extend my heartfelt thanks to all of you here today, and indeed all of our donors and supporters. You are how we continue to be able to be prepared to take advantage of being at the right place at the right time.
I've spent 50 years as a scientist doing medical research, largely at the medical school, then in Monsanto, and then the pharmaceutical industry. That research was really about dissecting and understanding of how does the life process work? What are the elements that really control normal function? And when we understand some of that, then we could see what's distorted in disease and how we could do a therapeutic approach. So when it was important to come and join the Plant Sciences Center, well, I came to think about that the tools at which we attack the problem are kind of the same. They're called cell biology or molecular biology or biotechnology or chemistry. And the dissection of the problem is similar. And then I comforted myself with the notion that a plant is really a person that doesn't move. Well, for 4 wonderful months, I've been in almost constant tutorial in evaluating science. And pretty quickly, I was struck by what does it mean not to move? That plant sits there and is attacked by bacteria, by fungus, by viruses, by nematodes attacking the roots, by insects and predators that want to eat the plant, by drought, by cold, by salt. So what has the plant done? It's been thousands and maybe millions of years of Darwinian selection to really optimize survival in an environment. And each plant, when you apply the tools of science has unique properties of survival. But now, plants weren't evolved for humans. We became users. And no plant satisfies all the conditions because of their environment. So much of the science here and in other places digs into the fundamental understanding of the unique characteristics of plants and how did they survive, and how do we learn from Mother Nature, and then how do we fool Mother Nature? Because in plants, we could go back and redesign plants for enhanced function, because you could use the genes, which are blueprints for the desirable characteristic, and insert them in plants and fulfill the highest expectations to improve food production, to overcome the stresses of deterioration, to use plants to attack the human condition, to use plants to attack the environment. So what I've done is, from the many things that are happening here, I've picked 4 kind of research areas just to illustrate. It's kind of, they're at the stage where the discovery operation is pretty much done, but I want you to see the science and how it advances and see how we've been able to take the genes and insert them where they are needed to have a beneficial outcome. First, I want to talk about 3 examples of where a plant's productivity is destroyed by fungal attack. Here there are 3 fungi, one in the US that destroys large amounts of wheat and barley, and also produces a substance which is toxic to people. Here are two fungi that attack corn. Here, ear rot, which is prominent throughout sub-Saharan Africa, not only has the toxin, but that toxin is a carcinogen. And the third is a global problem of fungi that attack and produce galls and release the product. Now, without belaboring the years of work, by searching in plants and viruses and other tissues, 3 antifungal genes that made proteins that protected the plants in their environment were discovered at the Danforth Center. Now, the proof of concept is, can we take these genes, insert them in a plant, expose them to the attack, and do they survive? Here is one example. Now, the plant Arabidopsis is the model system. It's the mouse for the plant sciences. In this case, they're going to be exposed to that fungus that attacks it and produces levels of this toxin. So here's a normal plant. Here's a normal plant that has no protective gene, exposed and inoculated with the fungus. You see the plant is destroyed with this high level of toxins. These three Arabidopsis plants, transgenic plants, have had the genes put in for one of these proteins called DEF4, and you see it lowers the level of mycotoxin, and the plants are survivable. So it's a laboratory experiment, it's a bench experiment. The next great challenge is move it into relevant crops, like in wheat, demonstrate the level of expression of the gene. When you succeed in the greenhouse, then you go to field trials. Another example of the fungal attack, here's pictures of a fungal ear rot. And in this case, two of the genes, now you could add, remember, seeds and plants are really information systems. This is about changing the program for information. And here they're programmed with two genes and exposed in the greenhouse and then in field trials in collaboration with North Carolina, where they were exposed to the fungal ear rot, and this is the worst case scenario. And here, when the gene was inserted, this is the best case scenario. Now, this is not yet optimum, and there has to be a lot of work to maximize the expression of the protective antifungal, and that's the ongoing work. And that was going at corn rhinrite, and then they have an antifungal protein that could go after corn smut. Bottom line, you can indeed take the lessons from antifungal proteins, insert them in plants, and really make an impact on deterioration. Next, I want to talk about, and Jim, there's a lesson here about a cell that you ask about. How do we use plants as factories? Now, in this case, the plant is soybean. You all probably raise peas and other legumes. They fix nitrogen, and they're high producers of protein. A soybean seed is 40% protein. So here's a cartoon of a cell. And the reason I want to show it to you is because how does it make protein? The gene is a blueprint that comes from the nucleus, and it manufactures a protein that moves through two compartments in the cell and gets trapped into a vesicle where it's degraded. So it has a very transient life. Some of the protein goes into structure, but some of it is degraded. Now, what Elliot Herman and Monica Schmidt did is they wanted to take better control. So they attacked two genes that changed the compartmentation of how the protein was processed and redirected part of the pathway such that now it didn't pass through the two systems that would process it, and in fact, it got packaged in these protective bundles. And in those bundles, the protein can't be degraded. So they diverted the protein, some of the 40%, into something that's going to be potentially useful. And now, if you peer into real cells with an electron microscope, here's the normal cell, and what you're looking for is these clumps of black cells. That's all new protected protein that then could be harvested. Why is that important? Well, not only now, on the one hand now, you could use proteins for the mass production in soybean of industrial level enzymes of importance that you couldn't otherwise. Let's divert into an environment. You all understand corn to ethanol, but that's corn that's not going into the feed crop, that's corn that's using agriculture. It has a profound impact. In that corn plant was the stalks that get thrown down. There's all this grass. Those plants have a sugar in a polymer form that's not usable, cellulose. What they're doing now is working out systems for the mass production of an enzyme, cellulase, that will degrade those polymers, save the food, and now use crops and use that as a source of sugar to ethanol. So we're dealing with the protein as a factory. We're dealing with it to generate critical things that affect the environment. We're fulfilling part of the objective of the Danforth Center to have an important influence on agricultural commerce and in St. Louis. And this was one of the first startup companies that started in the Danforth Center. Second example, now there's a few. Instead of the mass production, there are now pharmaceutical enzymes that are being produced in plants, which are so potent that they could be produced in greenhouses. In this country, human albumin and lactoferrin and other proteins are produced in rice. In Israel, there's a protein that's being generated that treats some of these rare genetic diseases. Well, those plants pale compared to the capacity of the soybean. And already we've been contacted by Washington University that wants to look at ways in plants that we could make novel antibodies that could be used in oncology. And the third thing is all the animal food. While they eat plenty of soybean, they don't have all the relevant amino acids, like they're lacking methionine. These folks could re-engineer the pathways. Now I'll switch to another kind of problem. What do we do about the disastrous impact of malnutrition? Not just our own improving our agriculture. And you've heard a lot about cassava. Cassava is this root plant. Here you're seeing it being harvested. Maybe 300 million people in Africa use cassava as a staple in their diet. The reality of cassava, it's about 60 or 80% starch, so you get the calories, but it's nutritionally deficient. And so if you eat those cassava, you'll have malnourished children. One example is the fact that cassava makes both beta-carotene, which is the precursor of vitamin A, and it makes vitamin E. But it's only 4% of the minimum daily requirement. In Nigeria, which is a target, about 23 million children less than 5 years old use cassava as their main source of calories. About 900,000 of them are disabled, and almost 34,000 of them die a year because they're deficient. So the inventions done here by a large group that Sam pointed out, with Dick Sayre and Claude and Nigel Taylor and Paul Anderson and Martin Frigine and really a large team of collaborators. Here we're going to take advantage of the plant that already makes vitamin A precursor, and there's a number of steps and a branch point. And the branch point goes either to vitamin E or to vitamin A's precursor. So if you really want to do something about nutritional deficiencies, this is a 2-gene strategy. You insert the gene that really drives stealing material from starch and drives the carbon flux. Now, what they learned, that wasn't enough, because at the branch point, it didn't go where you want. So it requires the second gene to drive preferentially, so you now make vitamin A while you don't steal vitamin E. Here's what a transgenic cassava looks like, which has had those 2-gene inserts. You could just look at the color and know the difference. So here's the control. Here's this very low level that are in normals. And these plants have a 30-fold increase in the expression of beta-carotene, which goes to vitamin A. And if those 5-year-old or less children eat that, they get 100% of their minimum daily requirement of the vitamin. Now, these are still early days. This is just an exploratory pilot trial that's done in Puerto Rico. Ultimately, to work in Africa, every part of the country has its own unique plant. You have to get the genes in the relevant geographic cultivar, and you have to get it stable. Well, we have now done the very first transgenic plant trial that's ever been approved in Nigeria, and there are plants there still not in the relative landform. And by later in this year, we want to put that 2-gene construct to go into the landform that they use, for example, in Nigeria. It's called TME7. Now, if you look at the rest of cassava, here you have plenty of the starch. It's deficient in vitamin A, deficient in iron, deficient in protein. Scientists here, both in the greenhouse, in the laboratory, and now in the earliest trials in Puerto Rico, have achieved the desired in a model system. They've achieved the vitamin E levels, they've achieved the necessary iron levels, and we've reached levels of protein that are usable. Along the way in these plants, in Africa, in Western Africa, in Nigeria, there isn't a viral attack load. In Eastern Africa, it's rampant. Again, and they never evolved the Darwinism for an antiviral approach. So there's a separate whole effort done in collaboration with a number of people and supported by Monsanto in a grant that allows the insertion of multiple strategies that will make cassava resistant to virus. And there's been some success in delaying the deterioration. What comes forward is a lot of science. You can't just grow the plant. You have to see what's the price to the plant. If the plant doesn't give you the same yield of roots, if the plant doesn't flower, if an African farmer who lives on this sees something wrong, so you have to really show that you don't lose the characteristics. We have to learn to stack multiple genes, and we have to get it in the right landform, and that will be in Kenya and Uganda and other places. So the main objective is within 4 years' time, our target in the big Grants Foundation grant is to have modified plants ready for wide regulatory trials by African farmers, some are being trained here and involved in Africa as well. Lastly, let's talk about the environment. You all know algae, and you probably do everything you can to kill it in your swimming pool. It's a microorganism, it's single cell. Its characteristic is that it uses sunlight and CO2 to make its energy. So it has other very important properties. Algae could double very quickly in size. Some species, 6 to 12 hours doubling time. It has a high level of lipids, of oils, and those oils will become the biofuel. And there are other programs here that work on oils, but in this group of people who are led by Dick Sayre, it's focusing on how to use algae. You see, it's not in competition with agriculture. You could see here, if you compare the leading crops that could be sources of oil, you could see algae under optimum conditions almost make 1,000 times higher oil yield and could use 800-fold less land area. Now, algae alone is not going to be the solution, but biofuels for jet engines, for diesel, and some petroleum. Now, the trouble with algae is if you went to the oil, it's not economical. It's off by a factor of 4 or 8. So there's a lot of science that has to go forward to change the dynamics of that. Now, the lists of possible problems are considerable, but some of the attacks are, how do we improve the delivery of solar energy and photosynthesis? There's work on what's the antenna, both at Washington U and here, for the capture of the sun. There's changing the genes so you could change the ratios of chlorophyll. There's work here and in other plants where you redirect how the algae makes oil and go to the most beneficial pathways. You could increase the amount of yield. So you got to increase biomass. You got to increase efficiency. You got to increase oil. You got to do it and learn how to extract without killing the algae and preparing the water. And the objective already, and that's led to another spin out, is this year there'll be a pilot plant in Hawaii using algae similar to this plant in Israel called Symbiotics, which is a partner with FICOL. This is next to an Israeli energy plant which is generating electricity from coal, directly diverting some of the carbon dioxide, trapping the carbon dioxide into the generation of new biofuel. Now, how does this happen? It really started in 2007 when the Taylor family awarded and created here at the Danforth Center the Enterprise Rent-A-Car Institute for Renewable Fuels. This is investing in the fundamental sciences that could lead in a renewable source of energy that really has alternatives to petroleum, and invest normally in the basic science. And as you saw in Sam's slide, that's generated multiple sources of funding from the Gates Foundation, from Department of Energy, and the wherewithal of this, which is so attractive, is they invest in the basic science that we need, as well as coming to practical applications. And it's an ambitious target to say, in 4 years, we're going to really be economically viable in this area. The other result is, in many ways, St. Louis has become a leading site, if not the leading site, for algae sciences in this country. There's one other thing about algae that really tweaks my old medical instincts. I think 60 million people a year get malaria. And there's another problem where we're going to use algae in the Sierra laboratory, and it was a Gates Foundation grant. You know that the vector is carried by a mosquito. The mosquito loves to lay its eggs in ponds, and here are the larvae, and they're on the surface of the ponds feeding on algae. Now the mosquitoes make an insecticide. However, it's completely broken down. So in this proposal, and it's broken down by an enzyme, this proposal, the algae will be engineered to make an inhibitor of that enzyme. So the mosquito's own pesticide will then be killed by these transformed algae. And that's going to have a trial in Mizzou later in the year. So in closing, how are we doing on the mission? Adding micronutrients where you'll influence thousands and millions of people. Using plants as factories to make biopharmaceuticals. Using plants to avoid the destruction of fungal attack and eventually of some of the other stresses. Some of these plants are going to facilitate the production of oils or starches for fuels. And then finally, what about the region? Well, there's 3 embryonic startups. You know, when the science moves to development, it physically moves from here as a separate enterprise. And there's at least 3 companies. One you saw the protein one you saw the algae one. There's another company emerging that will use the starch of cassava in this country instead of cornstarch. The second and important thing that you should realize is the Danforth Center has now developed a reputation that they really understand, because you can go into Africa where others haven't, many do, understand the development process, the regulatory process. In many ways, the Danforth Center becomes the go-to place for any academic who has a gene and wants to do a trial. And then you see the power of the algae sciences. So while scientists just follow their dream and the curiosity and hypothesis-driven, it's always on the horizon, how do we make this important and change the human condition? Thank you.
Well, thanks to Sam, thanks to Phil, and thanks to you all for being here. We can't stop now. We'll close with a quote from Winston Churchill. Man, he would say today, men and women, or women and men, have never sought tranquility alone. Nature drives him or her forward to futures which are different from those which it is his or her power to pause and enjoy. So we're now ready for comments, questions, challenges. Well, let's see. Yes.
Wow.
Ron?
Congratulations on an incredible decade of progress.
Well, thank you. Thank you. Well, I hope that when the report is made in 2020, people will say incredible progress in the last 2 decades. But if there are no other questions or comments, we will break a few minutes early. Yes.
What criticisms does this organization ever receive?
What you mean internal or external? You want to say?
We could talk about the societal attitude about genetically transformed plants. The world has changed. You know, it used to be enormous pressure on the Africans because of the Europeans not to take those plants. But they could see the vitamin A, and they'll be able to see the iron. So it's better, but it's hard. And the other hard part is we're doing trials in unstable places. So when we do a trial, we're trying to train African scientists. We're building some of the facilities. We're learning with the regulators how to do it. Now, in this country, take the soybean issue. Soybean in protein could worry soybean farmers. So there's a lot of work here in other plants, like camelina, where they've already gotten up to 6%. So if it's in sensitive areas, there's alternatives, or we could do it in the greenhouse. So we're really learning as we go forward. The experience of Monsanto has changed some of the dynamic, but it's always a challenge. So we're very cognizant of the responsibility of how to handle it, how to communicate, and who we collaborate with. It's changed so much because of the involvement of the Department of Energy. It's wonderful through Bill that there's an NIH equivalent now. You know, there never was plant sciences funding like there was in NIH. What is it, a factor of 7 or 8?
13.
13. What you don't realize is in order to have an institute and be attractive, it cost us maybe 52 cents on the dollar for every dollar of research to build those core facilities, the international program, the greenhouses. Well, the dynamic of that is improving now. And now that Howard Hughes, which was such a big supporter of medical research, so they're now engaged in part of the education process about plants and plant sciences. This is truly the cutting edge. If you waited just for conventional breeding, in another life I worked across the street in Monsanto where they wanted to increase yield. It took about 10 years to add 1% increase in corn growth. With genes, they could do it 7% increase in 1 year. So the impact has to earn its right, but it has to earn its right in things that improve the human condition. But that is an ongoing challenge.
I would just add to feel that, you know, as humankind moves forward, there are always corrections that are needed. You learn new things. Some of the things that you're doing are fantastically successful and helpful, but then they have complications that have to be changed. And another kind of criticism that I think is very valuable is internally from your friends, always looking for ways to do things better. And if you're not doing that, if you're not criticizing yourself, you stay pretty static. There's some other hands up. Yeah.
How do you balance the need for your commercial partners to protect their intellectual property with your interest in going out and developing science for the masses?
first of all, we're very careful because we're a nonprofit organization, and we're very cognizant and develop conflict of interest and even guidelines for starting up companies. If it derives from the science, we even have mechanisms led by a committee of the board led by Bob Virgil for always assessing that. After some embryonic period, they really have to move away. So they develop intellectual property. We have advisors. That's available in negotiation for licensing possibility. But all the science of development has to separate as it goes for-profit activity. So the bridge is the complement, but we're very careful about maintaining how do you write patents, who is part of it, and if it's for-profit, it's different than our venue.
And go ahead.
Well, one thing that Roger did from the very beginning, which was unique here at the center, is that from day one, he saw the potential conflict in our dual missions of commerce for commercial partners and feeding the world. So he put a stake in the ground, made a proclamation, which now we were the first, but many have followed. We will offer no exclusivity for our technology to companies. If you are Syngenta or Dow Agro or Monsanto and you want to have access to a technology, you can have exclusivity only for the commercial world. We retain the right to give it freely for the developing world. That was unique. The World Bank's Consultative Group and International Ag Research followed that, and many other institutions have followed that. It was very pioneering.
Thanks. Yeah.
When you modify the cassava, you change the color. What about the taste or the texture? Are you concerned about the locals accepting the new product?
The question was, what are the implications? That's part of the question. What is the price you pay? Now, those plants that you saw the cross-section of, there's a institutional review board application from Mark Maneri at Washington U. This is a regular plant. They'll be feeding experiments to even see, is it bioavailable and do you get the levels up? As part of the outcome, you have to measure yield, appearance. I mean, that's part of the regulatory challenge. So what you saw was early. It's, does it taste right? I mean, farmers will even be thrown off if it flowers more or if yield is late, and they know there's plants. So the ultimate success is it looks like, it feels like everything that they know, and it has all the characteristics. If it doesn't, you go back to the drawing board, because unless it's adapted by the people on the ground, but our objectives are even higher for them. We would like them to not only, they lose 30% just rotting on the ground. If we could optimize that, they'll have more than sustainable agriculture. They'll be able to be commercial, and we could change the economics on the ground. So we've got to get the properties right. And so this is early in the game. There'll be lots of field trials, lots of assessment of parameters. There's biosafety, you feed it to animals. There's a lot of steps to make sure it's nutritionally right and fulfills all the safety criteria.
Go ahead.
I just wanted to first of all say thank you. I mean, I think that that's the best thing we can say to all of you for everything you're doing. It certainly makes us feel very proud. But I know that it was so exciting that the governor came here with regard to Mosira, and I was just wondering if there was benefit in us taking some sort of a leadership role as citizens and obviously very proud stakeholders, if we could be of service to that for the state, if there's anything that you would suggest for us.
Well, Mosira is a way in which some states have supported their research, and it's the closest thing to think about it is it's not a TIF, but it's like a TIF in that you capture some of the taxes that come from increased business over a period of years. And Missouri has always been behind in funding its own scientific development and its sciences. And we're hopeful that this bill that's favored by the governor will pass in this session. And of course, anyone who can help us do that or let your representatives or your state representatives or your state senators know about it, that would be helpful. Thank you. Well, anyone else? Am I missing anybody? No.
One question. Thank you. One question. As you progress, human resources are going to be an inevitable need. And do you think the universities, not only, stateside, but around the world, are staying up with building the sciences and engineering that you're going to need, that the globe is going to need to carry these programs forward?
You know, algae is a good example. If you're really going to scale, it's a giant engineering problem. It's the transport of the fuels. The movement in big grants is for multidisciplinary. You'll need a lot of other kinds of skills. So here, our skills more often than not are modifying the genes. All that algae has to be extracted. It has to be in the right oil form so it can go into the refineries. Unfortunately, those oils, as they see in other of the fat-related research, are of the right sort to be biofuels. But it's engineers, it's chemists, it's going to be a lot of business, it's going to be a lot of infrastructure.
I would just add to that you know, in our modern society, everything kind of fits together with everything else, and you need to advance lots of things. But all that we hope to do in the future, all that we hope for our country, is going to depend upon education. It's going to depend upon education from preschool to PhDs and doing it well. And that's something that in parts we do well and in parts we don't, but we can all sort of favor improved education.
Dr.
One
Danforth.
more question?
Yes,
Okay.
one.
I can't
First of all,
see.
congratulations on the success that you've had. I know of your mission, that third mission I have a particular interest in, and that's making this region the hub or the Silicon Valley of plant science. How are we doing in that area, and what can we do more to make that happen?
Sam, you want to talk about some of the...
Sure. Alonzo, I think, first of all, it's something that we started years ago with a study that was done on the strengths of our region, the Battelle study, telling us that we should play to our strengths, and our strengths were a strong scientific base, great research at Washington University and our land-grant institutions, a Monsanto company. So we played to that strength. The Bridge Park leverages the strength of the Danforth Center, both of its intellectual capital and its equipment and specialized growth rooms and chambers that companies can rent if they move here. It's critical infrastructure. We as a team together work to try to add value to companies, because if we're going to build this cluster, the last thing that of importance really is the bricks and mortar. That real estate is a commodity. It's all those other things that we have to convince companies that we offer that others don't. A gathering, a networking, access to capital, partnering, and I think it's a long road, but we've had a good start. I had a meeting yesterday with Joe Schlafly, and he said that he was at a meeting, and folks are already talking about the progress that we've made here in St. Louis. So a lot of different pieces. I think we're moving in the right direction, and I think, you know, unless we stumble, we'll get there.
One thing to think about is, supposing you were starting a plant science company of some kind, and you're an entrepreneur, and you're thinking that down the road you wanted to sell your company and have it acquired so you would do well and make a lot of money. And you thought, where would I like to be? Wouldn't you like to be in Monsanto's backyard? So that's in a way how everything sort of fits together. Well, thank you all very much for coming. As you leave, we have a memento of this occasion, a green a coffee cup or tea cup that says Donald Danforth Plant Science Center. And on the other side, it says the right place, the right time. Thank you for being at the right place at the right time. successful.
Well,
Congratulations.
it fits so well.
It was wonderful.